The three letters, DNA, are deceptively simple, but what they represent is extremely complex. Deoxyribonucleic acid, or DNA, carries the molecular blueprint in cells that is the chemical basis of all life. Two complementary chains of molecules spiral elegantly around one another, concealing an extraordinary sequence of genetic information. And with every new cell that's produced, the DNA's code is divulged. The information is replicated in all its intricate detail to continue the cycle of life. This detail concerning DNA and other cellular molecules is at the nucleus of current molecular and biochemical research.
Welcome to the world of DNA. As an animated chromosome, I'll review the structure and the composition of DNA with you. Then together we'll look at how, when, and why the DNA molecule replicates. Finally, we'll turn our attention to the advances in DNA research and glimpse the future of genetic engineering.
So this is DNA?
Yes, Tanya. And this model here represents the culmination of the work of many different researchers and scientists.
It looks like a twisted ladder.
You're right, Corey. Its structure is a double helix. And these strands here represent the backbone of the DNA molecule. They're made up of alternating phosphate and sugar molecules. What about these molecules here between the strands?
Those are nitrogen bases.
A base from one strand bonds with a base from the other strand. The order of these base pairs is critical because it provides the genetic information that will eventually lead to all our structural and functional proteins.
So how does this DNA model relate to chromosome structure?
As one chromosome, I represent a small amount of the DNA found in each cell of the body. In humans, there are 46 of us, 23 from each parent. Just one of us contains between 50 and 250 million base pairs of DNA, about 17 millimeters in length.
But how does all of that genetic material fit inside the nucleus of a cell?
Well, Tanya, the nucleus of the cell is highly organized. The chromatin, or genetic material, is a long strand that can be coiled tightly and twisted like a rope into a small space, something like this. Really, a chromosome is one very large DNA molecule.
Each of the trillions of cells in the human body contains all of the millions of base pairs for the entire human genome, except red blood cells which do not have a nucleus. The total amount of information encoded in the chromatin, human genome, is 6 billion base pairs. The nitrogen base is only one part of a subunit called a nucleotide. Each DNA nucleotide consists of a deoxyribose sugar, a phosphate, and the nitrogen base. An enormous amount of information is encoded in DNA using only the 4 nitrogen bases: adenine, guanine, cytosine, and thymine. The different sequences and number of these base pairs along the polynucleotide strand is the genetic code, which provides for the tremendous variability of genes and their protein products.
We're actually going to see DNA. I can just imagine what Watson and Crick must have felt like when they discovered the double helix.
Yes, but they still hadn't seen the actual molecule. I mean, we know about the nitrogen bases, the phosphate, the deoxyribose sugar.
Let's get started. I have the calf thymus all prepared. I'm just going to place it in the blender.
Before DNA can be released from the nuclei of the calf thymus, the cell membranes and nuclear membranes must be broken down. This can be accomplished by breaking up the tissues into tiny bits, evenly mixed in solution. We're homogenizing the calf thymus tissues in a blender. Okay, now we have to fill these up to the neck.
Okay. This the first one.
Remember, they have to be filled evenly.
All right. About there?
Yep.
There's the last one.
Great.
Want to take that to the centrifuge?
Now we'll spin them for 30 minutes at 4,500 RPM.
Just wait until it's stopped completely. Okay, let's get these out. Take them back to the bench.
The cap thymus cells have come out of solution. We're going to throw out the liquid part and keep the cells. That's where the DNA is.
All right, we've got to pour out the soup name. Let's get the beaker, distilled water, and the caps. So you hold that beaker for me. I'll pour off.
The resulting homogenate contains proteins, carbohydrates, lipids, and nucleic acids. Chromosomal proteins stripped from the DNA and other cellular proteins must be precipitated.
All right, we've got the soup off. Now let's add the distilled water. Put the caps on those.
And what exactly was this shaking supposed to do?
It's supposed to get the pellet out, so it's easier to mix. All right, well, let's pour the suspension to that beaker. Go ahead. All right, now if you can add the distilled water till it's 50. Okay, now I'm just going to mix it really good. Okay. Now, I have to add some SDS detergent to break the cell envelope. Just going to take this SDS, use this plunger, two milliliters in there. And what we're going to do, take a glass rod and stir it up. All right, now we're going to layer it with ethanol. Okay, now we'll have you measure it with a graduated cylinder.
Of the components remaining in the homogenate, only DNA is insoluble in ethanol. So, when ethanol is added to the homogenate, everything remains in solution except DNA.
This has to be done slowly so that it layers. Stirring it slowly See now, you can see the threads forming around the stirring rod.
The molecular structure of DNA stays intact. It's a thick, stringy white mass.
DNA is a long chain of nucleotides. Each nucleotide consists of a 5-carbon sugar bonded to a nitrogen base and a phosphate group. The 5-carbon sugar or pentose in DNA is deoxyribose. Deoxy because it lacks an oxygen atom that is normally found in the ribose sugar molecule. The nitrogen bases are either a purine, a double ring structure, in the case of adenine and guanine, or a pyrimidine, a single ring structure as in cytosine and thymine. The phosphate is attached to the 5th carbon atom of the sugar and links each nucleotide to form a polymer or polynucleotide, the whole DNA molecule. This results in a backbone with a repeating pattern of sugar-phosphate, sugar-phosphate. The sequence of nitrogen bases on this backbone is specific and unique for every gene on a chromosome. But the story also has a twist. The double helix or spiral structure that Watson and Crick unraveled. You see, DNA consists of 2 polynucleotide chains. Sugar-phosphate molecules form the backbones on the outside of the helix. Nitrogen bases are paired in the interior of the helix. Adenine pairs with thymine, A with T, and cytosine pairs with guanine, C with G. The pairs are held together by hydrogen bonds, and it is these hydrogen bonds that cause the strands to coil, forming the helical structure.
If all the DNA in your body were uncoiled, stretched out, and connected, it would reach the moon and back 8,000 times.
Okay, so we have to understand how the specific sequence of nucleotide bases in a DNA molecule is replicated to create an identical copy of itself.
Doesn't DNA synthesis and replication have to occur at each cell division without error?
Yes, it does, Corey. And that's one of the points of what Crick called the central dogma of molecular biology. The dogma is based on the idea that information flows from DNA to messenger RNA during transcription to proteins during translation. The flow of information is at the very core of life.
Okay, so let me see if I got this straight. Information is stored in DNA, encoded in RNA, and then decoded to form cell products, right?
That's right.
All right, I think we're ready to make proteins.
Not yet. First, we have to replicate the DNA.
Okay, how does that get started?
Remember the cell cycle, Tanya?
Yes.
Well, DNA is replicated during the S phase of interphase of the cell cycle. After a period of cell growth, the DNA condenses into thick strands that we associate with chromosomes. The condensed DNA cannot be read.
Once cell division is completed, the DNA becomes uncondensed. It's now called euchromatin, and it can be read. Let me use this model of DNA here to further show the structure of the molecule.
DNA polynucleotides have a beginning and an end, just like sentences do. At one end of the molecule, a phosphate group is attached to the 5th carbon. This is called the 5' carbon of the sugar deoxyribose. So this is the 5' end of the molecule. At the other end of the molecule, a hydroxyl group extends from carbon number 3. So this is called the 3' carbon of the sugar deoxyribose of the last nucleotide of the DNA molecule. So this is the 3' end of the molecule. Similarly, within the DNA molecule, a bond between adjacent nucleotides is formed between the hydroxyl group of the 3' carbon and the phosphate group of the 5' carbon of the following nucleotide. So this strand runs in a 5' to 3' direction and is called the leading strand. The opposite strand runs in the 3' to 5' direction. So these two strands are anti-parallel to each other. And this is necessary if the DNA is to be read properly.
I see. Are we ready to replicate yet?
Almost. Here's what has to happen. The DNA has to untwist, like this, and then the nucleotides of each antiparallel strand have to be copied simultaneously. This requires a coordination of more than a dozen enzymes and other proteins.
DNA replication is extremely rapid. In prokaryotes, up to 500 nucleotides are added per second. It only takes a few hours to copy the 6 billion bases of a single human cell.
The process of DNA replication is extremely accurate. Only 1 in a billion nucleotides is incorrectly paired. DNA replication begins at special sites called origins of replication that have specific nucleotide sequences. 2 specific proteins bind themselves to these origin sites. There are many origin sites to facilitate the replication of large amounts of DNA. The proteins untwist and separate the parental DNA strands. The beginning of each new strand of DNA is initiated by short sequences of nucleotides called primers. When the primers are in place, enzymes called DNA polymerase begin the synthesis of new DNA by linking nucleotides to a growing strand. The nucleotides align themselves to the template of the parent strand and form hydrogen bonds. Adenine binds with thymine and cytosine binds with guanine. The strand grows in the direction of 5' to 3', since new nucleotides can only be added at the 3' end of the growing strand. Now, continuous synthesis of both strands of DNA at the replication fork is not possible because the sugar phosphate backbone of the complementary strands run in opposite directions. They're antiparallel. So DNA polymerase can only elongate strands in the 5' to 3' direction. The problem is solved by the continuous synthesis of one strand, the leading strand, and the discontinuous synthesis of the complementary strand, the lagging strand. The lagging strand is produced as a series of short segments called Okazaki fragments, which are each synthesized in the 5' to 3' direction. The many fragments produced are linked together by an enzyme, DNA ligase, to form a continuous chain. Now, DNA synthesis is considered semi-conservative. The new DNA is made by using the original DNA as a template. One parent strand and one new daughter strand appear in the final product.
James Watson and Francis Crick proposed the semi-conservative model for DNA replication. They predicted that when a double helix replicates, each of the 2 daughter molecules will have one old or conserved strand from the parent molecule and one newly created strand.
So, before a cell divides in mitosis, the DNA must be replicated.
Yes, and precisely replicated. The information in a gene is determined by the number and sequence of bases that comprise it. There can be anywhere from 2,000 to 1,000,000 base pairs in a given gene sequence.
So, every single nucleotide base must be in the correct place.
Right. What happens if there is a change?
Any change in DNA is called a mutation, and this sequence here is certainly mutable. For instance, the base pair of cytosine and guanine could be changed by thymine and adenine. That would change the sequence of the DNA. But mutations are a source of new variations and essential to the process of evolutionary change. However, not all mutations are advantageous. Some can alter the function of a gene, which could lead to genetic disorders or cancer.
Or couldn't there just be a mistake in the replication process?
Yes, a mistake in the copying of DNA during cell division could cause a normal gene to become carcinogenic.
The study of recombinant DNA has triggered many research advances. New techniques have opened up the study of the molecular details of gene structure and function. Some of these techniques include PCR, polymerase chain reaction, where many copies of different DNA sequences are created. Molecular cloning, where exact copies are made of genetic material. and DNA sequencing, where automation and computers are used to analyze nucleotide sequences. These techniques have been applied to the Human Genome Project, an ambitious research project with the goal of mapping the entire human genome. DNA exists not only in the double helical form found in humans, but can also be linear, circular, supercoiled, single-stranded, or right-left twisted. As new information comes to light, our understanding of DNA is modified and improved.
Mistakes in replicating DNA are bound to occur, but we don't all have cancer. Are the mistakes corrected?
DNA repair is essential for correcting errors and repairing damage. Did you know that DNA is the only macromolecule in the cell that is repaired instead of replaced? What enzymes do you think are involved in DNA repair?
Well, DNA polymerase must have something to do with it.
There are many different polymerases. This group of enzymes perform a multitude of functions associated with DNA replication. For instance, if an enzyme spots an error in base pairs, what it will do is remove the incorrect base and replace it with the proper one. Another enzyme, DNA ligase, is also involved.
The message must be the same, or the genetic information will be changed.
There are more than 50 different DNA repair enzymes. They can reverse a change, remove a base pair, and refill the gap. Accidental changes in DNA could be caused by exposure to reactive chemicals, radiation, X-rays, or ultraviolet light.
Yeah, like skin cancer from UV and sunlight.
And radiation from X-rays.
That's why you're covered in a lead apron when you have your teeth X-rayed.
Right. The cells continuously monitor for accidental errors and repair most of them. However, as you get older, the repair system becomes less efficient. And it should be noted that if the damage is severe enough, whether you are young or old, cell division and cell growth can be adversely affected.
Like severe radiation in nuclear accidents like Chernobyl.
Yes, I guess even repair enzymes can't deal with those kinds of errors.
No, they can't. The study of restriction enzymes has enabled scientists to learn a lot more about DNA sequencing. Restriction enzymes cut the DNA into smaller fragments, and these fragments can be used to determine the exact sequence of bases in a DNA molecule. This knowledge makes it possible to manipulate genes.
Is that what they call genetic engineering?
Yes, that's it.
Our main application is to genetically improve, in some cases, the quality of the oil in the canola, and to use canola for a model system to understand the genetic makeup of organisms. We use canola to insert a gene that might be foreign to it and produce what we call a transgenic plant. And we do this process in order to use the plant to produce a certain foreign protein, which may be an industrial application, or it may be a food enzyme application, or in fact it might be a pharmaceutical product. If we look at these plates here, we'll see that we have the canola seeds in this sterile petri dish on a media that allows them to start to germinate, and we can see here that the seeds are germinating. And eventually what will happen is we'll get 2 cotyledons that will come out from this germinating seed, and we can cut the cotyledon, as we see here. Each of these leaves are cut on the tip, and we dip the end of this cotyledon or leaf into a solution containing what's known as agrobacterium, which is a bacteria that will transfer DNA into a plant. And we have genetically modified that bacteria to contain our gene of interest. The agrobacterium's function is to transfer the gene from its DNA into the plant DNA, and through a series of tissue culture steps, we can generate a transgenic plant. Now, we don't always know in fact, that the plant is transformed until we can do some further testing, because we have to prove whether or not our gene of interest went into the plant. And so we have to do a series of tests. We might take some leaf material off of a plant like this, and we can extract the DNA from that leaf tissue by grinding it up and putting it through a series of buffers and solvents, extract the DNA from there, and then be able to prove that our gene that we wanted to put into the plant is in fact in the plant. And then we have produced what we call a transgenic plant.
What are the regulations governing this transgenic research?
We have very rigorous controls. As I said, we have done 2 years of field trials now, but in our first year we had to actually cover our crop with tents and fences around them in order to prove that nothing is going to outcross out into the normal canola crop. And as the more of these field trials we do, we feel that we're able to produce or to provide more information to the regulatory agencies that in fact what we're doing here is done in a very safe and controlled manner.
Only 3% of all the DNA in the body, about 100,000 genes, is involved in producing proteins. The purpose of the rest of the code is not really understood.
Can't imagine what those other types of DNA would do.
It's hard to believe that our life processes are controlled by molecules.
Yeah, like DNA, a macromolecule coding for an RNA message.
Yes, it all starts here in the nucleus and extends to the ribosomes in the cytoplasm. The genetic code, manifested in the DNA molecule, is shared almost universally among living organisms. The only difference between a human gene and a bacterial gene is in the number and sequence of base pairs. In fact, experiments have shown that bacteria can translate genetic messages from human cells, and human cells can translate bacterial genes. Mitochondria and chloroplasts have their own DNA, containing genes to synthesize specific proteins. These essential metabolic proteins are found in a multitude of diverse organisms. This fact gives testimony to the evolutionary principle of shared genetic roots among the living organisms of this planet.
In this program, you have explored the basic intricacies of DNA structure and replication. Understanding DNA's double helical structure with its sequencing of nucleotides and hydrogen bonds between the bases is key. Equally important is understanding how the DNA double helix uncoils prior to replication, and how RNA primers and DNA polymerases work to build new strands of DNA during replication. Unraveling the mysteries of DNA structure and function has greatly advanced the science of genetics and our understanding of the molecular basis of life. It has also opened doors to new possibilities in the field of genetic engineering.